I'm Marie Bernard, the chair of the Women of Color Committee of the Trans NIH Working Group on Women in Biomedical Careers. And I'm happy to welcome you to the NIH Director's Wednesday afternoon lecture series. Happy New Year. Although we didn't plan it this way, today's lecture on the topic of pain, the first for 2021, serves as a nice metaphor for this moment in time, considering the painful year we've had and the hope for relief in the coming months as a result of our leadership and creativity and dedication to science. And now about today's speaker. Dr. Omowumi Sadik is a distinguished professor and chair in the Department of Chemistry and Environmental Sciences at the New Jersey Institute of Technology in Newark, New Jersey. She is also the director of the BioSensors Materials for Advanced Research and Technology Center, called BioSMART for short. Her research areas include biosensors, surface chemistry, electrochemistry, and nanostructured conducting polymers, with an emphasis on developing innovative technologies to improve human health, food safety, and the environment. Dr. Sadik holds several US patents in biosensors and sustainable nanotechnology. Her inventions help drive changes in many areas, such as the early diagnosis of cancer, genetic testing, environmental analysis, and pain management in hospital patients, which we will hear about today. Dr. Sadik earned her undergraduate and master's degree in chemistry from the University of Lagos in Nigeria. She earned a PhD in chemistry from the University of Wollongong in New South Wales, Australia, and then followed this up with an NRC postdoc position at the US Environmental Protection Agency. Until recently, she was a professor of chemistry and director of the Center for Research in Advanced Sensing Technologies and Environmental Sustainability at SUNY Binghamton. She has also held appointments at Harvard University, Cornell, and the Naval Research Laboratory. She assumed her current position at the New Jersey Institute of Technology in 2019. Dr. Sadik's work was named by the Chronicle of Higher Education as one of 10 research projects across the world which could keep society safer. She is a fellow of major scientific academies, including the African Academy of Sciences, fellows of the Royal Society of Chemistry in the UK, and the American Institute of Medical and Biological Engineering. She has received the University of New York Chancellor's Award for Research and Creative Activities and the NSF Discovery Corps Senior Fellowship. The title of Dr. Sadik's talk today is Smart Biosensors, a new modality to objectively quantify pain. Dr. Sadik, thank you for taking the time to be with us. We really look forward to a great lecture.
Thank you very much, Dr. Bernard. I'd like to thank all of the world's organizers and the Women of Color committees for the invitation and for organizing everything for today. I am pleased to be able to give this lecture despite what's going on. I have been to the NIH campus a couple of times, and I was really looking forward to actually visiting again. But other than not having the lectures at all, I'm still pleased that we can do it virtually. And so thank you so much. So today I'm going to talk about how we are using biosensors to measure biomarkers of pain and ultimately creating a model for objectively assessing the level of pain that people are going through. And so let's see if I can, okay. So we're doing this work at the BioSMART, as you've already heard from the introduction. And our BioSMART is the acronym for Biosensor Materials for Advanced Research and Technology. Our building in the Newark is on the Life Sciences and Engineering Center at the NJIT campus. And currently we have a host of graduate, undergraduate, and postdocs who are involved with the BioSMART. But we also have a selection of distinguished faculty who are contributing in one way or the other to the BioSMART Center. And I have a selection of some of them here. Dr. David Bada is a computer scientist who works with us on machine learning and data analysis. Dr. Kevin Belfield is an organic chemistry professor and dean of science. Dr. Sadik Basure is a chemical engineer who works on biosensors and microfluidics. Dr. Hao Chen is an analytical chemist. His expertise is in mass spectrometry. Dr. Simon Garnier is a biological scientist, and he looks at swarm intelligence, using smaller organisms to understand how we can develop smart biosensors. Dr. Mengin Lee is a biological engineer, and Dr. Samitra is an analytical chemist. So together, we have a model of BioSMART where we will take a look at organisms like the slime mold, which does not have any nerves, and yet it can perform certain sensory units and creating what we learn from this organism to actually develop robotic sensors. But for the purpose of today, we're going to focus on pain biosensors. So biosensors, I'm going to briefly explain what they are and how biosensor devices are leading to wearable biosensors. And then we'll focus on pain. Pain is very difficult to define, but we will try. We'll look at the transduction of pain, the modality of pain, and then the core aspect of the presentation will focus on the assessment of pain as well as the management of pain. In the assessment of pain, we'll look at the biosensors that we've developed. We have 3 generations of the biosensors and how we tested this out in human populations. And then in the management of pain, we'll look at how we're designing new flavonoid derivatives that can potentially serve as the inhibitors for some of the pain markers. And then I'll conclude. So biosensors, what are they? So typically biosensors are small size devices. They would have things like a biological component like enzymes, nucleic acids, lipids, receptors in cross connection with a transducer. Okay? The role of the transducer is to convert the recognition of this biological agent for the analyte into a measurable signal. It could be analytical signal, it could be electrochemical or optical in nature. And then the biosensor can be used in many different forms. There are wearable sensors, which is where the state of the art is in the field, to other small handheld devices that people use at home. A very good example is the glucose biosensor that diabetic patients will use to check the level of glucose in their blood. Typically, the response is quick, and within seconds to minutes, you should have it's kind of an answer to what you're looking for. And so biosensor would go beyond that. And as I said, we can take a look at the components of a typical biosensor on this slide here. The analyte is typically what we're interested to detect. Let's say glucose. We want to know the glucose level in the blood, or we want to know the amount of biomarkers that are responsible for pain in the blood. And the receptor will be in cross connection with this transducer. And your receptor could be the biological agent of interest. It could be cells, mammalian cells, it could be bacterial cells, it could be a portion of the cells like the enzymes or antibodies. And once the analyte binds to the receptor, something is triggered within the transducer. And the transducer could be electrochemical, or it could be optical or other forms of transduction mechanisms. And then the signals are either amplified or sent directly to a type of readout. Typically, a signal is obtained. So let's say that we have glucose sensor, which is the most common that a lot of people can recognize, although biosensors extend way beyond that. The glucose device, the glucose monitor device is an electrochemical sensor. And essentially what it's doing is it looks at the glucose level. There's an electrode. Electrode is a conductor, like a platinum or aluminum. And so on this electrode, which is going to be the transducer, you would have a very small layer of an enzyme called maybe glucose oxidase. So the glucose oxidase is the receptor, and the electrode is the transducer. And the analyte of interest will be the glucose. And so you can look at the glucose biosensor. It was developed in 1960, in the 50s, late 50s, where we look at the reduction of oxygen to water. And typically, Clark and others decided that they can actually use the understanding of the electrochemistry to create a sensor to monitor glucose. And so the first commercial glucose analyzer came out in 1973, and I believe was developed by Medisense. It was a huge instrument. Today we have very tiny sensors that you can barely even see. And from there, others developed. We have the GlucoWatch, we have the test strips that's now ubiquitous in homes. And of course, in the 2000s, with the development of nanotechnology, we developed, we now have the nano biosensors. Those are based on enzymes. As I said, biosensors can also be used, based on antibodies or other receptors. And so we can have antibodies to other to immunoassays. We have other types, even DNA based. But today, we've gone beyond just the strips and devices to have wearable devices. Wearable devices are getting very common for things that can look at this in the sweat. You can have devices that will look at sodium levels, potassium, ions in general, even glucose and lactase and many things, all in one device. And you can also have flexible sensor arrays where you probably think this person is wearing a watch, but what they're probably looking at is monitoring different aspects of their body. So today we have wearable biosensors ranging from even eyeglasses that would be able to monitor lactate or glucose. You can have a smart contact lens that would also be able to look at the glucose level and a host of others, so many different applications of this. So wearable biosensors that can send information to telemetrically, even to physicians, so that you don't really have to be physically there to monitor what's going on. There are others. This is all biomedical in nature, but there are environmental sensors, there are food sensors, there are sensors for virtually everything you can think of. So to talk about pain, pain is common. Pain is whatever the person tells you they're experiencing. And over 40% of Americans visit the clinic every year due to either chronic or acute pain. And it's a multi-billion-dollar industry. Pain is one of the reasons why people don't come to work for the most part, losing productivity. Pain is one of the reasons why the lifestyle change, is really could be lowered if, if someone has chronic pain, the quality of life actually is affected. And the likelihood is, if we take a sample of people in a single room, most people, you probably have at least a couple that will be feeling pain at that point in time. And so how do we address the problem of pain? Typically, of course, there are analgesics that have been developed over the years, and they are useful, except that it is very difficult to, over time, control this, because the more of analgesics we take, the higher is our level of tolerance for some of this. And so you might then need to go to higher levels of painkillers like the opioids and morphine. Is actually one of the reasons for the opioid crisis that we have currently. And so how is pain transmitted? How is pain measured? How is it managed in general? And what are the challenges of transmitting of pain? So before we can do that, we have to define it. And as I said, pain is very difficult to define. Pain is what that person tells you they're feeling. But the International Association for the Study of Pain came up with a definition. And they define pain as unpleasant, sensory, or it could be emotional pain. You can take a look at people and think, well, you don't seem to be in pain. But they can convince you that indeed they are feeling pain. And it could be actual or it could be perceived. But in most cases, there's some sort of damage to the tissue. And so the definition is difficult, so is the quantification of pain. So if we're able to quantify pain, just like we're able to quantify the glucose level in the blood, then I think that we've achieved a great deal. But we're very, very far from there. And what I want to show you today is what we have done, at least from our group, to look at how to develop tools, biosensors, for objective assessment of pain biomarkers. So before we get there, how do we feel pain? Why do we feel pain? We feel pain because messages are being sent to the brain. There is a some level of, okay, so we would have maybe there's a site of injury, okay? The whole body is filled with receptors, nerve receptors that are sensitive to pain. So if there is any penetration, so the receptors are sensitized and messages are sent through the nerve, to the central nervous system, to the appropriate region in the brain, and we know exactly that we feel pain, okay? There is a reaction. If I put my fingers on a hot stove, messages sent quickly, and I remove my feet. That's the reaction. I have pain, in essence, is there to protect us. Pain is there to warn us that something is wrong, so that we can take appropriate action. But in some cases, and in most cases, pain becomes very chronic, that we really don't have any, the body itself cannot respond appropriately. And so there has to be some form of intervention. And so pain is complex. It is multidimensional. And in most cases, it's not a condition, it's a symptom of something else that's happened. So we can classify pain into two: either acute, something happens, you fell, you remember you were doing something happened, so it could be acute pain, or it could be chronic over time. There's this constant feeling of pain, maybe due to some sort of underlying diseases. So the difference is the duration of the pain. So currently, how is pain assessed? There are several scales, assessment scales for pain. It could be based on the verbal intensity scale, where, no pain to very, very, very severe pain, or worst possible pain on the scale. It could be based on a visual analog scale where they'll give you some sort of, some scale, and they tell you, on the scale of, this number, where do you stand? It could be numeric, or it could be based on what is called the Wong Baker face scale. Usually they use this for children. But the problem with many of these assessment scales is that they tend to be very, very subjective. In most cases, in some cases, the whoever is taking the test can over-exaggerate. They can exaggerate the pain that they're feeling, or they may even under-report the pain. Let's say somebody is unconscious. You have no idea. They're in trouble, but you don't know how much pain they're in. Or they're children, and they're not able to even verbally express their pain. So how do you help them? So that is why we need objective assessment scales, okay? So currently, pain can be managed through some sort of intervention to the transmission or the transduction of pain. In the 60s, there was the pain gate control mechanism that allowed us to understand that when there's any injury to any part of the body, the nerve endings, the pain receptors are activated, and messages are sent to the brain, and there is a response. So, but then they realized that, look, what if there's an injury and we're able to create some sort of to interfere with the transduction of the pain to the brain, then we're not going to feel the pain. And so we can use the interruption of the signals by maybe massaging the spot or the area where the pain is being felt, or we can use local anesthetic to control, to affect the pain. Or you can even use physical therapy or electrical stimulation. There's so many ways we can control and interject that transference of the pain. But either way, all of these could be subjective as well, because I can continue to take more of those anesthetics, and my body can develop such resistance to it that I need more and more and more. And over time, especially in situations with chronic pain, people become addicted to painkillers. So we need objective analysis of pain, not just by looking at trying to understand the underlying cause of that pain, but to look at the intensity as well. So if we understand the intensity and we're able to quantify pain, then it is possible for us to find unique ways to actually address it. So NSAID, the non-steroidal drugs, they're widely available, and these are common ones. But there are some other non-selective or selective enzymes that when you have them, they are typically indicators of certain types of pain, or inflammation, or other types of pain. One class is the cyclooxygenase enzymes. The cyclooxygenase 2 in particular is not native to human body. Once you start getting COX-2 within the blood, it's an indication of something that is going on, perhaps inflammation. So cyclooxygenase 1, there are 3 isotypes of this: COX-1, COX-2, COX-3. COX-1 is native to the body. In fact, it is part of the normal functioning of the body. However, once you start having COX-2, then there's trouble. Others are the inducible nitric oxide synthase. You can also have prostaglandins. There's so many different types of biomarkers that one can actually use as indicators to develop these types of biosensors. So another problem is not just the intensity as well as the cause of the pain. Painkillers in most cases, there's no guidelines. We are working with physicians, a host of physicians, and they said there are no guidelines relying on biomarkers. And so there's no objective treatment currently. NIH came up with the HEAL initiative. The HEAL initiative is the helping to end addiction long-term initiative. And the idea is to be able to develop a whole class or slew of biomarkers that can serve as indicators for different types of diseases. They could be genetic, they could be proteins, and then develop instruments or devices that would help to objectively quantify and then define the guidelines to how to address this problem. So with the NIH HEAL initiative, it goes from lab to all the way to clinical use, discovery, analytical validation, clinical validation, and then qualification. But before the NIH HEAL initiative, we have been developing sensors for pain. And so what I want to now shift to is what we have done. We've worked on this project for over, I would say, 15, 16 years. We believe that if we understand the biochemical mechanism of pain transduction, then we can develop objective tools. That is the hypothesis. Understanding the biochemical mechanism of pain transduction so that we can develop objective tools to measure it. For example, how much pain is this man feeling and how can we help? Well, if you go to the hospital, they're going to give you the space. And as we've already established, that is not objective. So let's look at the biochemical mechanism of pain transduction. There's been a lot of research. Biochemistry of pain markers have been scattered for different diseases. But I'm going to focus on prostaglandins, okay? So if you focus on, I'm not going to bore you with much of the chemical structures, but it's probably the only chemical structure you see in this talk. But let's focus on this. So we have the arachidonic acid, these are lipid molecules on the lipid membrane, and the conversion of arachidonic acid into prostaglandin G and H takes place by catalysis of the cyclooxygenase 1 and 2. The reaction takes place within 2 minutes, and ultimately that leads to the formation of other types of markers down the line, like the PGE2, PGF, PGI, and all of that. But overall, the cyclooxygenase essentially stereochemically removes the 13 pro S hydrogen and control the stereochemistry of oxygenation in this place. This is the oxidational step. So if we can focus on this slide at this stage of the formation of PG2, which is only a few minutes, we can monitor the level of COX-2 that is present, or the ratio of COX-1 and 2, so that we can tell before it gets into this downstream molecules where everything gets very complicated, then we should be able to develop at least some objective markers for pain. So the arachidonic acid metabolites are important in the objective measurement of pain, and in particular for inflammation. So as I said, the arachidonic acid, they're produced from the phospholipids and the cell membrane. Now we have steroids, for example, that can interact or block the formation of these arachidonic acid, okay? And once you don't have that, let's say you have the arachidonic acid itself, it can form prostaglandin, G, H, but in between you have the cyclooxygenase 1 and 2, and maybe even 3, that are inhibitors of COX-2. So there are many inhibitors of COX-2. Aspirin is one. So many others. There are several problems with the inhibitors of COX-2, as we'll talk later. Another direction we could go is to look at the lipo-oxygenase proteins as well. And that could take us through all of these different directions. So let's say we follow the cyclooxygenase pathway. With the cyclooxygenase pathway, you can form the PGI2. These are causes of the vasodilation. They will inhibit platelet aggregation. You can have the thromboxanes. Those will cause vasoconstriction and promote platelet aggregation. And you can go down the lane from there. So you can also look at other types of mediators or biomarkers. And one of them is the inducible nitric oxide synthase, iNOS. Okay? So the iNOS is very important for the nitric oxide, and nitric oxide is an important molecule for nociceptive transmission of pain. So now we have a series of biomarkers that one can actually look at. You can look at COX-2, you can look at iNOS, you can look at prostaglandins, you can look at arachidonic acid, you can look at a host of other neurotransmitters and create a whole class of markers that could be detected and quantified, and that could have direct correlation with the level of pain. So the objective then of our work, the long-term objective, is for us to develop an objective scale based on the fundamental biochemical mechanism of pain transduction. That's our objective. We want also to be able to predict by linking our objective scales with machine learning, okay? But in the meantime, we've developed sensors, biochemical sensors, to monitor COX-2, prostaglandins, and many others. And we've also developed molecules that can inhibit some of these proteins. So if we're going to develop sensors that will not just detect, but also be able to predict, that will have physicians make predictions of the level of pain, then we need to be able to use machine learning. And machine learning, of course, is getting very popular. We've used machine learning for decades in my group, not just for pain sensors. We've used this for the energy-related molecules, anthrax. We did that for anthrax and many, many others. But the key thing is that with machine learning, you have data, okay? You have data, but within your data, you want to be able to extract features, important features that would give you an indication of what is going on. And of course, create algorithms that will help you to make predictions. So it's a combination of data science, mining, classical programming. And if you look at machine learning in literature, in scientific literature, they're really bogged with a lot of mathematics. And we need to be able to translate this into very simple languages that ordinary person can be able to use. So here's what we've done. We've developed multidimensional biosensors where we're combining the signals from the biosensor device with 3D facial imaging, okay, as well as what the patient tells you. Don't forget, from my definition, pain is what the patient tells you they're feeling. And so we cannot ignore what they tell you, right? And so we have to take into consideration the self-reporting from the individual. And then you look at the psychological component as well. So this project is a multidisciplinary, multi-campus information, in fact, multi-nation, as you will see from the result I'm going to show you. And we've looked at a whole segment of the American population, as well as Turkish patients as well. And I'll tell you why we ended up going to Turkey. It was just coincidental. However, we're also looking at, as I said, development of non-opioid based analgesics. So we've developed a suite of biosensors based on different mechanisms. And the reason we need a suite of biosensors is because you cannot rely on only one thing. The biosensor from one will be used to validate or to multiplex another signal. And so the four that we've looked at is, one is based on standard enzyme-linked immunosorbent assay, ELISA. Our biosensor, this one uses electrochemical mechanism. We call it MEG. This one uses surface plasmon resonance techniques with, based on SPR. And then the instrument that was actually developed for my lab based on fluorescence, we call this the UPAC. And so we've combined the signals from all of these different platforms for the same objective. So ELISA, I won't go through the details. One method is immunoassay technique that is multi-steps, and this is how we use it. We just basically have antibody. We have a COX-2 here, human recombinant COX-2 enzyme, and we have antibodies to that. One raised in human, one raised in goat, and we can generate signals. So I'm not going to go into details. For the SPR, we have another technique, again, to look at the same information, where we have a direct recognition of the antibodies to COX-2 or iNOS, and we measure how it binds and creates equilibrium and dissociation for the protein. And the third type of sensor that we've developed is based on the electrochemical detection, where we have nanoparticles on silver, and we use antibodies to COX-2 or iNOS, and we do the recognition of the protein, and there is some sort of changes in the electrochemistry. And the last technique, or the last sensor that we've developed, is based on the UPAC, the ultra-sensitive portable capillary sensor. This instrument was developed in my group. It was originally funded by the Department of Defense. And the idea was, in fact, I was in Naval Research Lab way back in 2000, maybe a year before then. And we wanted to test a simple concept. Take a simple glass. You can see the glass here. It's a very simple glass. Let this glass serve multiple functions. Let it be the transduction device. You already know the transduction now, based on the definition we gave about biosensor. Let it serve as the solution, like it's fluidic as well. Let it serve as also the surface whereby you can attach antibodies or enzymes. Anyhow, we use this, and we tested this for some of the toxins of interest to the Navy. In particular, we use what is called this SAB, Staphylococcus enterotoxin B. There are different classes of these proteins. And a very small percentage of this protein is like a snake venom we can kill. And typically, so the Navy was looking for a way to detect this. So we tested the concept, and it worked very well. But we used a very vast, huge instrument in the lab. And then 9/11 came, and we were funded by the DOD to miniaturize it, to make this portable. And that's what led to the use UPAC that you're seeing. This UPAC is kind of protected by so many different patents. Anyhow, we decided to use UPAC as well to compare the result with ELISA, with the SPR, and with the MED, electrochemical device. Here's another version of the UPAC. And this one here, we can do a lot of other, rather than using laser here we use LED. And it's a lot cheaper than trying to do LED. And the third generation of our sensor is not even portable, but it's handheld, okay? With the ubiquitous nature of cell phones, now we use this on paper. We have a simple program. This was actually funded by Bill and Melinda Gates Foundation for us to develop a paper-based test strip for low cost smallholder farmers. And we tested this in Jamaica. That's another sensor that we're developing, but we've also used this for our pain markers. So let me show you some of the results. So typically we can calibrate this sensor. Here's an example for the SPR. When you have the COX tool and you have the control, there's a big difference. For iNOS, we use a similar kind of concept for ELISA, and you can do a nice sigmoidal response as the concentration of iNOS is changing. And so it works very well for iNOS as well. And so we did comparison of all four. As I said, we are using a multi-dimensional sensor arrays. So we're comparing the electrochemical, the SPR, and the fluorescence UPAC method with the ELISA, which is the gold standard for immunoassay. And we looked at different parameters, and we found that ELISA will take 3 days, your med is a few minutes, SPR will be half an hour, and the UPAC is 5 minutes. Automation, we can automate. We did the version of UPAC, one version of it was automated, and it worked very well for the analog of interest. Now, we also linked our sensors and integrated this with machine learning. And essentially, as I said, we're collaborating with computer scientists, pain management specialists, physicians, and psychologists. So they will call us to the hospital to collect blood samples. And my students will go there, and the nurses would do all the IRB paperwork, and they would give us blood samples. However, we found this project at that time was being carried out by Idris Yazgan. And Idris is from Istanbul. And it would take them a long time. The samples were not coming, and it was getting frustrated. Before I jumped the gun, this is how we linked to machine learning. So we'll create expression models for different faces. So here you're seeing the 7 expression models of different facial expressions, okay? You can have neutral, angry, disgust, fear, happy, sad, surprise, okay? So these are all different expression models. And then you will, the system will have this 3D camera that will project. So once they call us to come and take blood samples, the computer scientists will take the cameras as well. So they would take pictures of the patients after their consent. And they'll project the light onto the subject and capture the shape of their face precisely using this synchronized digital camera. And then each of the facial expression, here is an example of the geometric models that of the happy face, okay? And here is another expression of the surprise face, okay? So you come up with different geometric models. And then each of these will now, you will now project them at different angles. So the cameras are set at various angles to give the optimum configuration. Once that is done, they would take the, so they would train the program, the software program with different data. So you have this trained data. They use what is called the hidden Markov model, HMM model. So the idea is that the hidden stage is X, for example, and then you compare that with unknown, and as you begin to change different dimensions of the visual expressions, and you train the software with different algorithms rating to track, to adapt, and then you create this geometrical face spatial expression. There are several stages of this. You collect the samples, you extract features, and then you use this model to train. And then you can now ask it to make predictions. So there's recognition, and then it gives you the result, and then it recognizes. For example, what does it mean? For example, if you have human nose, we have hundreds of sensors in the epithelial region of the nose. And then you have the mucus with proteins. And so the sensors are not specific. They are selective. They change continuously. And so over time, you train the human mind to recognize the smell of coffee, the smell of different things. So because they're not specific, so we're more or less creating, we're simulating the human sensory system by creating arrays of these sensors, and then using this algorithm to do the brain to train the sensor to recognize different levels of the pain receptors. So we did pilot study, and we want to test, we collected just few samples to see even does it work in real human lab thing after we've done all the lab testing. We created all the scales. 0 is no pain, 1, feeling some pain, but no disturbing, but disturbing you, strong pain and things like that, okay? For 15 patients, this is the first time we did that. 15, we said, well, we found there's a correlation for COX-2. You can do a nice calibration. And we also found that it correlated with the numbers provided by the patients. And so we were very excited that this is the way to go. That COX-2 is actually a good model to test our sensor. But then we were disappointed as we began to look at the largest sample set. In phase 2, we now decided to get more samples. So we collected close to 200 samples, and most of them were rejected based on certain criteria that we selected. We're going to the ER, and some of those reported unknown pain. Some of them had inflammation, some had direct accidents. But we now looked at each of these numbers. We used our sensors to detect the COX-2 level in their blood and the iNOS levels in their blood. We compared our result with the ELISA method, and use our 3 types of biosensors: the SPR, the UPAC, and the MAD, the MED. So we then fed the result into the machine learning, the classification model, to do a survey of how does it really, is there a correlation between the numbers that we're getting and what the patient is telling us, as well as what the facial expression, the 3D imaging is providing. And so we found that most of these patients had taken very heavy opioids, really, before coming to the hospital, which basically changed the result. We had to create a new, it's not even 1 to 5 now. We did what is called the modified scaling because it was completely off the scale. We used the iNOS. So we compared the result for serum levels in the iNOS relative to the serum, the COX level in the serum, okay? And we could see that the levels of, first of all, we need a number. What is a good number for iNOS? Nobody has that in literature. We have to establish that. What is a good number for the COX-2? Again, we have to establish that as well. But we're seeing some sort of correlation between the two. So here's what we did. So the levels of pain of 0 to 5, the number of patients that recorded each of these. So the one with 5 is about 35, 4 was 24, and so on. Then we found the level of COX-2 because we have to define that level. And so ND is non-detected under the experimental condition. So for those with 0 pain, the levels were between ND to 105 nanograms per mil, parts per billion. For those with 1, it will be between ND to 125, and the highest for number 5 is now 3,800 nanograms per mil. So we have some numbers. We have some numbers. So now we know what level of COX-2 should we expect if somebody is not feeling any pain at all. But over time, this level should go up. And we expect that anybody with 3,800 nanograms per mil should be about pain level of 5, okay? And we did the same thing for the iNOS. Without 0, no pain, we found that the iNOS levels are much higher than the COX levels. So in general, the COX levels are very low, but iNOS is giving you like 6,000 nanograms per mil. So we have a scale. So I'm going to summarize what the findings are, because this result was really, there was a lot of result. We found that for most of the patients that we took, most of them took painkillers before coming to the hospital. They're on very high levels of some of these opioids. Upstate New York is really bad with this. And so even if we have a scale, the fact that the patients have already been on very high levels of these painkillers is already changing the scale for us, okay? So we had to create what is called a modified pain scale for those who've taken NSAID before coming, for those who've taken, many of them will not actually tell you that they're taking opioid, but many of them had taken very heavy narcotic drugs like fentanyl. It was really, really bad. So we give them high level because the pain is already affected by the narcotics that they're taking. Now, coupled with the fact that the number of samples that we're getting is actually very low. So this is the levels for the COX and for the iNOS. The key thing to take from this table is that it supports our hypothesis that we can use COX-2 and iNOS as prognostic biomarkers to objectively quantify pain. We have a scale now. We have some level to expect. So for our first one, for the phase 1, where we took, hundreds of samples from upstate New York, we're finding that the self-report is actually being affected by the fact that some of the patients are taking very high levels of painkillers. 83% of the patients who reported pain also show the presence of COX-2 in their serum. That's a very good thing. It means that we have a high level. So COX-2 is actually a useful marker in this respect, okay? We also found that 53% of the patients showed the presence of iNOS. So about half of them. In fact, for certain diseases we found, for lung cancer, for example, the levels of iNOS was off the scale, okay? And then so we created the adapted level of pain. So we cannot use the standard 1 to 5 or 0 to 5. So we created adapted levels for the COX-2 and the iNOS. And then when we did that, we found that there's a moderate relationship between the level of this biomarker COX-2 and iNOS and the level of pain that people are facing. And lastly, we found that the intake of very strong painkillers will affect the quantification of the pain. Okay, so my student who was working on this for his PhD was Idris Yazgan, and Idris wanted to go to Istanbul to take more larger sample size. He felt that first, most of the patients that he will be looking at don't have access to high levels of opioids. Secondly, he thought that he could get more samples. So he was supported. He took the UPAC with him to Istanbul and spent months collecting samples from many, many patients. So that became our phase 2 level of study. Again, we targeted COX-2 and iNOS as our marker. And our goal is to look at the intensity and the type of pain. So he went ahead and did a lot of recruitment. 57% of the participants were women, 43% were male. And this is the age range, this is the weight, smoking, and non-smoking, and then alcohol. So we did the medical history. In fact, unlike in the US where we won't have access, as long as the names of the patients and the identification factors are removed, the physicians, even the director of the hospital, was very, very helpful. The data was provided for us. They exclude all of the identification factors. So we got a lot of data. For the Turkish patients, out of the 588 that were invited, we found that 73 were not eligible because maybe this improper, they didn't fill out the form very well. There's some, these were excluded. And then we found 515 eligible patients. And of those 515, we monitored the level of COX-2 in their blood alone. We looked at iNOS alone, and then we looked at the combination of two. In fact, we also looked at the ratios of COX-1 and COX-2. And we found that we can use the standard pain scale of 0 to 5 according to the standard operating procedures. The blood samples were collected. We use our ELISA. We also use our instrumentation for comparison basis. Here's what we found. We found that even though the literature provides valuable information that the presence of increased COX-2 and iNOS can provide, but nobody knows how much. They just say if there's increasing COX-2 level, there's some level of inflammation or maybe cancer, but we don't know exactly what this level is. This study provided numbers, quantitative numbers for that. We're able to find that for the serum, if the serum level of COX-2 and iNOS range from between 3 to 6,000 over in nanograms per mil, parts per billion, then we can use that as a scale of 0 to 5. We also found that for the iNOS, we also found the scale for the iNOS. We found that for most of the Turkish patients, we looked at over 1,000 people. With the heavy levels of COX-2, there is a very high level of inflammation and even some very serious diseases like cancer. Okay, so we're beginning to see some correlation here. We found that the average levels of iNOS varied. Anything below 250 nanograms per mil is like no pain at all. But higher level, you know that something is going on here. Heavy inflammation, there's some pathological conditions here. So the overall conclusion for the phase 2 study was that a total of 74% of the patients showed the presence of COX-2. So again, is now re-emphasizing, confirming what we found with the American subset, that COX-2 level is significant for inflammation and very serious pain levels. And so it can be used as a marker. And 78% of the patients show the presence of iNOS as well. So both the American cohorts and the Turkish cohorts confirmed to us that indeed those two markers that we selected are really, really significant. So I don't know how much time I have, but I can basically quickly go over the pain. How much time do I have?
We are actually kind of at the point that we would want to do some Q&A. So if you could wrap up in the next minute or so, we have several questions that have come in.
That's good. That's excellent. Thank you. So we're doing pain management. We're developing non-opioid analgesics, and we're using derivatives of flavonoids. COX-2 is been linked to cancer and many other types. There are so many inhibitors present, like most people recognize Vioxx, which has been removed from the market, Celebrex, Bextra. And there are so many side effects, and that's why they were being removed. We are looking at all these molecules as alternatives. And in fact, in particular, we made this in our lab, this phosphorylated one. And these are derived from fruits and vegetables. And you can actually buy some of these, like quercetin, over the counter. They're being targeted as supplements. The nutraceuticals, they are not actually regulated in some ways by FDA, but you can get arpeginine as well. So these are some of the molecules that we've made in our lab as analogs of inhibitors for COX-2 and iNOS. And these are all of the chemistries. I'm not going to bore you with that, but we've developed this new class of flavonoids, and we've shown that they actually they inhibit. We've done in vitro studies to show that they inhibit the COX-2 and the iNOS. And so I will sort of summarize that, give an overview. I've given you an explanation of the pain definition as best as we can. We know that pain is not, you cannot define, it's complex, and it's what whoever is feeling it can tell you. We know how pain is transmitted. We know the current methods of assessment of pain. We know that, but we have developed biosensors of multidimensional to monitor specific biomarkers of pain. And we've correlated the levels of these biomarkers to the pain that people are feeling. We've looked at different cohorts, some very small scale that were completely linear as expected. We've looked at the larger subset of American patients. And then we've looked at the larger set from the Turkish population. So in summary, I've shown you different 3 generations of biosensors that we've developed, the UPAC 1, UPAC 2, and also the handheld. I don't have a lot of time to go through the mechanism of how this works, but it works. We're using machine learning to train the sensors to be able to predict the levels of pain that people have based on the levels of the biomarkers that we develop. And there's not a lot of time to actually talk about the management of pain, but the take-home message is that biomarkers provide a means to objectively quantify pain. There's still a lot of work that we need to do to test this in many, many, many populations, but there's no doubt that works, and our sensors also work for that. And I would like to finally acknowledge the people who have been doing the work and some of our collaborators, all of them across from different institutions, the funding agencies that supported the work we've been doing, so many students, current PhDs, and many from my former institution. And of course, you can tell that this is a pandemic age of lab pictures with everybody in mask. Thank you so much. I'll be happy to answer your questions.
Thank you so much, Dr. Sadik. Beautifully, beautifully presented. And as an aging researcher, I'm thinking immediately, this is great technology that could be used in non-communicative individuals. We often have the question in someone who has severe dementia, what's going on? Are they really experiencing pain, et cetera? There are several questions that came in, and we really only have about 6 more minutes. So I'm going to give you a couple of those questions. One of them from Dr. Clayton, Director of our Office of Research in Women's Health, was that she was glad to see that there were roughly equal numbers of men and women for your studies. The question is, would you expect to see differences in normal iNOS and or COX-2 levels? And would you expect to see any difference in the relationship between COX-2 and iNOS to pain levels by sex, given that pain conditions are more prevalent in women generally?
That's a very good question. We do expect to see, we've looked actually at the gender differences for COX-2 and iNOS. We're seeing the levels are different. We've looked at, yes, that's the short answer. Yes, we do. We expected it, and we found that it was indeed the case.
Very good, thank you. And I forgot to remind people that the CME number for this is 31290. Again, CME number 31290. Next question. In terms of using this technology as a feature to assist in potentially determining a treatment for a condition, how do you account for sensitivity to pain? Say the levels of the biomarkers are similar across groups of people, but they subjectively feel very differently. How do you factor that in?
This is the question that people ask we could not. In fact, the way we started the work was just to prove that this actually works in people. And because it was not funded by NIH or, we got funding from SUNY Binghamton, SUNY system. It was a very limited number. Each of the patients that we recruited, we had to give them money. And so we could not really ensure that the demographic of the patients were really covered. We simply were looking to make sure that at least this works. Certainly, that will be the next step for us to look at that fine detail.
Okay, thank you. Another question, you use facial recognition, and there have been issues about facial recognition not appreciating varying populations. Were you able to have a great deal of heterogeneity among those people whose faces you used, or is that the next stage that you all are anticipating?
Again, that will be the next stage. We understand that those differences are there, and unless we are able to recruit and make sure that the patient population is really representative of the population itself, we cannot make that assessment. But we have not done that. We recognize that is a possibility, but we haven't done it.
Very good, very good. So Dr. Sadik, I want to thank you again. Marvelous presentation. I know that we have you lined up to meet with some of our postdocs, so we'll give you a moment to catch your breath. And hopefully, once we're beyond this pandemic, we'll actually get you onto campus to come and visit. So thank you so very much.
I want to thank you so much for the invitation. I've really enjoyed interacting with you guys today, and thank you very much.
Thank you.